Alright, hey everyone, so today we're going to be covering the Lac Operon. So let's start off with the basics. What is an operon? So an operon is a group of genes with a single promoter, typically found in prokaryotes. So there's multiple different types of operon systems. You have inducible systems, repressible systems, and constitutive systems. In inducible systems, that is a type of operon regulated by an inducer molecule that acts as a substrate. What does that mean? So in the example I like to give is the Lac Operon, which is consistent of three genes, LAC-Z, LAC-Y, and LAC-A, that are involved in the metabolism of lactose. So when lactose is present in the environment, this operon, which is normally turned off, becomes turned on due to the binding of lactose to a repressor protein that's binded to an operator sequence of the operon. That in turn causes it to change shape and deattach from the operator sequence, allowing for RNA polymerase to transcribe the genes needed to produce a protein that will break down lactose. So the operon is normally repressed by the Lac repressor protein, and when lactose is present, it binds to the repressor protein, changing its shape, yada yada, in the example here. So an inducible system is essentially just an operon system regulated by an inducer molecule. And in the case of the Lac operon, the inducer molecule would be lactose. For repressible operon systems, that's a type of operon regulated by a repressor protein that binds to the operator and inhibits transcription. And the example that I like to give of that is the tryptophan operon, which is consistent of 5 genes that are involved in the biosynthesis or the making of the amino acid tryptophan. The tryptophan operon is normally transcribed when there is low amounts of tryptophan in the environment. So this amino acid, if it's present in the environment, it would not be energetically favorable to make more of it since we already have some in the environment. However, when there is a low amount of the amino acid tryptophan in the environment, this operon is then turned on and allowed to transcribe given proteins that can make the amino acid with tryptophan. So normally, for this operon, in conditions of high tryptophan, the operon would be repressed. You would have no transcription occurring. So this is operon. So repressible operons are just operons regulated by a repressor protein binded to the operator, not allowing transcription. On the other hand, you have constitutive systems, and that's a type of operon system that is transcribed continuously, regardless of the presence or absence of a substrate or inducer molecule. So for this system, the given gene is always going to be coded for. It's going to be transcribed continuously. And the example I like to give is the LAC-I gene in the Lac operon. This gene encodes for the repressor protein that controls the Lac operon, and over time, the repressor protein in the Lac operon can begin to wear down and become dysfunctional, so you have this constant making of new repressor proteins. And that's an example of constitutive expression. Within these operon systems, you have types of control: negative control and positive control. Negative control is gene expression occurs unless it's shut off by a regulatory molecule. So an example I like to give of that is going to be the repressor protein in the Lac Operon. So normally gene expression would occur unless it's shut off by a regulatory molecule. In this case, the regulatory molecule would be the repressor protein. It's stopping the transcription of these genes. So that would be an example of negative control. On the other hand, you have positive control. And positive control means gene expression occurs only if a regulatory molecule aids RNA production or RNA transcription. So for positive control, the example I like to give is the CAP or CAMP binding complex that aids RNA transcription in the Lac Operon. So positive control is the aiding of transcription by a regulatory molecule or an activator. Now, these terms can apply to both inducible and repressible systems depending on the mechanisms relating to transcription. So in both of the examples, I related it to the Lac Operon. It has aspects of it which have negative control and other aspects which have positive control. Going on to Lac Operon genes, we have Lac Z, Lac Y, and Lac A. Lac Z encodes for beta-galactosidase. Lac Y encodes for permease. And Lac A encodes for transacetylase. Those are examples of structural genes, or genes that code for RNA or protein that are not regulatory located in the operon. Now, going on to the regulatory genes, we have the Lac I gene, which encodes for the repressor protein. Now, it should be important to mention that this Lac I gene is not a part of the operon. As we see in this image below, we have this right here. And this is all what is included in the Lac Operon. We have the promoter, the operator, Lac Z, Lac Y, and Lac A. We see outside we have the repressor gene. So it's a regulatory molecule for the Lac Operon, but it's not actually a part of the operon itself. All right, and now we have the promoter sequence. And the promoter sequence is responsible for initiating transcription. It's where RNA polymerase binds. We also have the operating sequence, which is involved in regulation of transcription. And it regulates transcription by being the area where the repressor protein binds. And structural genes, just genes that code for RNA or protein that are not regulatory. Now, the repressor protein or the repressor gene is going to encode for a repressor protein that looks something like this. This operon's also, operator should have a little area for the repressor protein to bind. So normally, the repressor protein would look like this, in which you have an area right here in which it can bind to the operator. And you have an area up here which lactose can bind. In this operon, normally what would occur is you would have the repressor protein bound to the operator. And then, if you were to have the addition of lactose in the environment, lactose would then, let's say this is lactose, lactose would then bind to the repressor protein on this side, changing the shape of the repressor protein, causing it to deattach from the operator sequence. And that would then, once it deattaches, there's nothing that can inhibit RNA polymerase from transcribing the rest of the genes. So that would allow for transcription to occur. All right, in the Lac Operon, we have a variety of elements. We have cis-acting elements and we have trans-acting elements. Cis-acting elements are DNA sequences located on the same DNA molecules as the genes they regulate. And an example of that would be the promoter. Another way I like to think of this is cis-acting elements are stationary. So, darn it. Cis-acting elements are stationary. So we see we have the promoter right here. This is a cis-acting element. The operator is also an example of a cis-acting element, and both of these are stationary. They cannot move. On the other hand, we have trans-acting elements. Trans-acting elements are proteins that can bind to DNA at specific sites and can activate or repress gene expression. And an example of that would be the repressor protein. So the repressor protein right here, this is an example of a trans-acting element. And what makes that differing from cis-acting elements is its ability to move. Now, what makes these so different is the fact that if we were to have two genotypes, we have two, for example, DNA strands, and they both have a Lac Operon. If we were to have, let's say this codes for the I, that's terrible. Here we have the I gene, here we have the promoter, P, here we have, they both look like circles. Here we have P, here we have O. And on this gene, we have the other I. Here we have the P, here we have the O. Let's say that this right here was mutated, or the P right here was mutated. Oh no, let's say the I was mutated. If this I was mutated, and this resulted in a repressor protein that was unable to bind to the promoter to the operator sequence. So it looked something like this, in which has no area to bind to the operator sequence right here. However, this right here produced a gene or repressor protein that was able to bind to the operator sequence. This is a regular working repressor protein. The difference between cis-acting elements and trans-acting elements is that this is trans-acting, it's able to move. So this can move from this DNA strand all the way to this DNA strand and take the place of this faulty repressor protein. Whereas these cis-acting elements are stationary, they're unable to impact any mutation this may have. If this had a mutation here, or if this had a mutation here, these right here would be unable to impact them because they're cis and they're stationary and unable to move. Alright, going on to Lac Operon elements, we have the promoter and the operator, which are both cis-acting elements, and we have the I gene, or the repressor, and that is a trans-acting element. So mutations on the operon elements can cause a variety of effects on gene expression. Mutation in the Lac I gene can result in faulty and altered repressor proteins. So if we were to have an I+, I+ means that it's a normal repressor protein that is going to result in a regular working repressor protein, in which we have an area for RNA polymerase, in which we have an area to bind to the promoter, and we have another area that binds to lactose. If we were to have I-, that would result in a mutant repressor protein, and this repressor protein would be unable to bind to the operator. Sorry, I made a mistake. This right here, it would be able to bind to the operator. The promoter is where RNA polymerase binds. Alright, so in this repressor protein, it's unable to bind to the operator. However, it's still able to bind to the repressor protein, or bind to lactose. Now, regardless of if it binds to lactose or not, it cannot bind to the operator, meaning that since it can't bind to the operator, there's nothing stopping RNA polymerase from transcribing the rest of the genes. So it's going to result in constitutive expression. If we were to have an IS, that would result in a mutant super-repressor protein. So in this repressor protein, we have an area for binding to the operator, but we don't have an area to bind to lactose. Since it's unable to bind to lactose, it's going to remain binded to the operator, and nothing is going to be able to change its shape to move it. So it's going to remain binded on to the operator regardless of if lactose is present or not. That's going to result in repressible expression. It's not going to be able to express any of the genes. Alright, if we were to have a mutation in the operator, OC, that is going to result in a change of the shape of the repressor protein binding area on the operator. So normally, where it would look like this, it would then switch to having a shape in which the repressor protein is unable to bind. Since the repressor protein is unable to bind, there's nothing going to be inhibiting RNA polymerase from transcribing the rest of the genes. That's going to result in constitutive expression. If you had a mutation in the promoter sequence, in which the promoter sequence normally would allow RNA polymerase to transcribe the genes, now we have a mutation in which the promoter sequence will not allow RNA to transcribe the genes, and that will result in no transcription at all. RNA is what transcribes the genes and it's what expresses the genes. If RNA is unable to transcribe and express the genes, then you won't have gene expression at all. So that's going to result in repressible systems. And now there's a hierarchy that I like to use. This hierarchy, you have the promoter on top, in which the promoter, if you have a mutation in the promoter, you cannot transcribe any genes regardless of any of the other mutations. Secondly, underneath that, you have the operator mutation. If you have a mutation in the operator, that means the shape of the repressor binding area is changed, in which any of the repressor proteins will not be able to bind to it, resulting in constitutive expression. IS is going to be higher than the regular working I, and then I minus is going to be the last on the list. Alright, so here are some images. Here is an example of if we had I minus. Remember, I minus is going to result in a repressor protein that is unable to bind to the operator. So in this case, the repressor protein is unable to bind to the operator. We see that it can't bind, so there's nothing going to stop RNA polymerase from transcribing the genes. So that's going to result in constitutive expression. Here we have a mutation in the operator sequence, in which there's a change of shape in the operator sequence, not allowing the repressor protein to bind to it. Since the repressor protein is unable to bind, nothing is stopping RNA polymerase. RNA polymerase is going to transcribe all of the genes. That's going to result in constitutive expression. If we were to have a mutation in the IS is going to result in a super repressor, meaning that the repressor protein is not going to be able to bind to lactose. That means it's going to remain binded to the operator sequence. Since it's going to be binded to the operator sequence, and it's not going to be able to detach, since lactose cannot bind to it and change its shape, it's not going to allow for transcription to occur, and that's going to result in repressed expression. Also, here's a note. F apostrophe is going to indicate a plasmid that has introduced another genotype into the organism. So transacting elements on a plasmid can make up for faulty transacting elements on another DNA strand or whatever, on another genotype. Since remember, transacting sequences, elements, they're able to move. So a transacting element, such as the repressor protein, is able to move from one genotype to another genotype, assuming that it's in the same organism. All right, so an example would be right here. If we have IS, P plus, O plus, Z plus, Y plus, we have a mutation in the I that's going to result in a super repressor. Super repressor means that you have no area for lactose to bind, so it's going to remain binding to the operator, and that's going to result in repressible system. All right, on to the example underneath it. Here we have, for this genotype, IS, a mutation on the I, P plus, O plus, Z plus, Y plus. On the plasmid, we have I plus, P plus, O plus, Z plus, Y plus. We know that on the hierarchy, we have IS above I plus. So this right here is going to result in a repressor protein that takes preference or prevalence over the repressor protein that works. And remember, since this is a transacting element, the repressor protein here, which is going to look like this, is going to be able to impact the genotype that it's located on, and it's going to be able to move and impact the other genotype, meaning that this is also going to result in a repressible system. All right, on to these examples. For example A, part of the problem is already solved. In order to produce beta-galactosidase or permease, beta-galactosidase is the Z gene, permease is the Y gene. We see that. So right off the bat, the way that we would solve this is, if we want to see if the genotypes will be able to produce Z, we have to look at the Z gene first and foremost. Here we see on this genotype, we have a working Z, a Z plus. A Z minus or a Y minus is going to result in a non-functional protein being a result, so we would not include those. Onto the other genotype, we see we have Z minus, meaning that this cannot produce a working beta-galactosidase protein. Now, let's look at the cis-acting elements with regarding the Z plus. We have an OC and a P plus. OC is second highest on the hierarchy, meaning that any of the transacting elements are not going to have an impact. This is going to be expressed, so that's going to result in Z or beta-galactosidase being expressed or being transcribed. Going on to the Y gene, looking at this genotype, we see we have a Y minus, meaning it's not going to produce a functional Y protein. Right here on this genotype, we have a Y plus, meaning it will result in a functional Y or permease. Looking at the cis-acting elements, we see we have O plus and P plus, so those are going to work normally. Now, looking at the cis-acting elements, we have I plus and I minus. On the hierarchy, I plus is higher than I minus, so that's going to result in a regular inducible system. Inducible meaning that when lactose is present, transcription will happen. This gene, that's permease, it's coded for. These genes are meant and coded for the sole purpose of being able to break down lactose. If no lactose is present, then these genes will not be coded for because their purpose is to break down lactose. You wouldn't need to code for genes to break down lactose if no lactose is present. And this is also an inducible system, so we would have no lactose, we wouldn't have the transcription of permease in areas or environments with lactose. We would have permease being resulted. Going on to part B. For beta-galactosidase, let's look at the Z genes. Right here we have a non-functional Z. Here we have a functional Z. Let's look at the cis-acting elements. Here we have OC and P plus. Right off the bat, OC is going to take prevalence over everything else, apart from P plus. So we know we have OC. OC is going to result in constitutive expression, so that's going to be plus and plus, regardless of lactose or no lactose. Going on to the Y gene, here we have a non-functional Y. Here we have a functional Y. Looking at the cis-acting elements, we have OC, but we also have a P minus. P minus is the highest on the hierarchy, and that's going to result in no gene expression, since RNA polymerase is unable to bind to the promoter sequence. So that's going to result in no transcription in any of the environmental factors. On to portion C. For beta-galactosidase, looking at the Z gene, here we see we have a functional Z. Here we have a non-functional Z. Let's look at the cis-acting elements. We have a plus and a plus. Now let's look at the transacting elements. Here we have an IS. Here we have an I plus. IS is going to take prevalence over I plus since it's higher on the hierarchy. IS is a super repressor, meaning that it's going to repress the transcription of the genes. So we're going to have no transcription occurring for beta-galactosidase. Going on to Y or permease. We have a non-functional Y, and now we have a functional Y. Looking at the cis-acting elements, these are both working. So we have to return back to the transacting elements. Remember, IS takes prevalence over I plus, and it's transacting, so it's able to move, so it's going to impact this genotype, so it's going to result in no gene expression of Y as well. For D, we see that we have, for beta-galactosidase, or for Z, we see we have a working Z here. We also see we have another working Z here. Looking at the cis-acting elements, we have a working operator, a plus, and a plus. Looking at this one, we have a plus and a plus. Now we have to compare between both of the transacting sequences. Here we have an I minus, here we have an IS. IS is higher. IS here is higher on the hierarchy than I minus. That's going to result in super repressed expression, meaning that there's going to be no expression occurring. And since we have IS occurring, it's not going to allow for the transcription of any of the genes for either Z or for Y. On to E. Looking at Z, we have a Z plus right here. We have a Z minus right here. Looking at the cis-acting factors, we have OC and P plus. Since we have OC, that means we're going to have constitutive expression of the operon for the Z plus, so that's going to result in transcription and transcription. Looking at the Y, here we have Y minus and here we have Y plus. Let's look at the cis-acting factors. We have a working promoter, unworking operator. Now we have to go to the transacting system elements. Here we have an I minus, here we have an I minus. I minus results in constitutive expression, meaning it's always going to be expressed. So we're always going to have expression of Y. You guys can work on these right here, but the answers for these ones are going to be plus, plus, minus, 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 plus, minus, plus. All right, now, here we have one more element or aspect of the Lac Operon. So even when the operon is transcribing the genes, the rate of transcription is relatively slow. So two regulatory molecules are introduced and facilitate RNA polymerase binding to the promoter. We have CAP and we have CAMP. CAMP, also known as cyclic AMP, is a small signaling molecule that is produced from ATP being synthesized by an enzyme, adenylate sialase, I think. I don't know how to pronounce it. I like purple. CAP, on the other hand, is the CAMP receptor protein. And that regulates transcription based off of CAMP levels. Alright, so this is just essentially an added layer for transcription. It helps increase the rate of transcription. It does not affect if transcription will occur or not occur. So lactose is being broken down into a smaller molecule, glucose. And glucose is the preferred source of energy. If glucose is present, then there is no need to readily break down lactose. Lactose, if it's present in the environment, it's still going to be broken down but it won't be broken down at a fast rate, as compared to if there wasn't any glucose in the reserves. So, I guess to make it more understandable, the levels of CAMP. All right, so CAMP is synthesized from ATP by this enzyme adenylate sialase. And so the levels of CAMP in the cell are regulated by many factors, and the main factor that regulates it is glucose availability. When glucose is present in the cell, it binds to the enzyme adenylate sialase, which is what makes CAMP, and it binds to that enzyme, not allowing it to make any CAMP. When glucose is absent, adenine acetylate activity is able to occur at a higher rate since there's nothing binding to it, so it's able to break down ATP and make more CAMP. So when glucose is present, we are going to have when glucose is present, we're going to have low levels of CAMP. However, when glucose is absent, we're going to have high levels of CAMP. Now, both of these affect transcription rate, not if transcription will occur. So, if glucose is absent, then we will have the CAP molecule right here bind with the CAMP, and that will result in the CAP-CAMP complex. And that will bind to an area on the promoter region, and that will help increase RNA binding to the promoter region, and it help increase transcription. So that is essentially the Lac Operon.